Okay, so my research question was, is there a correlation between the density of gastropods and the density of Fucus spiralis beds in Point Pleasant Park? Fucus spiralis is a type of brown seaweed found in rocky marine intertidal zones, and I originally thought it was Ascophyllum nodosum until I looked closer. You can distinguish it from Ascophyllum because it does not have air bladders, only receptacles, which are reproductive structures at the tip of the branches, and those can look similar to air bladders. This is a picture I took, so hopefully you can see the difference. I used gastropods as a general guild, and I mainly saw common periwinkles, but I did see the occasional whelk as well. I've also included Dalhousie's official land acknowledgement. Dalhousie University is located in Mi'kma'ki, the ancestral and unceded territory of the Mi'kmaq. We are all treaty people. So for field work, I went out during low tide to get maximum exposure, and I used a 50 centimeter by 50 centimeter quadrat randomly placed along a 10-meter transect line. And I also split the quadrat into halves, which was randomly generated. I did 6 replicates for a total of 6 different beds, and I recorded temperature, salinity, and the GPS coordinates of each bed in case I needed to come back or anything like that. I counted and recorded the number of holdfasts and gastropods within the quadrat half. And then for my statistical analysis, I did a regression analysis to determine if there's a correlation between the densities. Okay, so here are my results. That is my regression analysis. It had an R-square value of 0.8697 and a p-value that was less than 0.05 at 0.006668. The y-axis is the gastropod number, the x-axis is the holdfast number. As you can see, it's a negative slope, and the average ocean temperature between the beds was 12.96 degrees Celsius, and the average ocean salinity was 39.58, and I believe that's parts per million. So here we're gonna interpret the results that we just saw. So based on the R-square value, the density of holdfasts explains 86.97% of the variation in gastropod densities and is a good indicator. And based on our p-value being smaller than 0.05, it means there's a small possibility that the differences are due to chance. And the negative slope means an increase in holdfast results in a decrease of gastropods. So my results conflicted a little bit with other peer-reviewed sources. However, it was difficult to find studies specifically on Fucus spiralis, whereas other types were more popular, such as Fucus vesiculosus. And it's pretty closely related to Fucus spiralis, but still not the same. It was much easier to find research in tropical areas or on green seaweed rather than brown seaweeds. Most of the peer-reviewed articles I found had a positive relationship between gastropod densities and vegetation. For example, a study was conducted in Indonesia comparing the density of gastropods in seagrass meadows with an R-square value of 62%. However, another peer-reviewed journal I found said that abiotic factors such as temperature, exposure, salinity influence the distribution patterns of gastropods on rocky shores, which is where I was. And the reason I believe I got the results I did was because the areas I sampled with really thick densities of Fucus spiralis, such as the quadrat I showed on the method slide, has little space on the rocky substrate for the gastropods to actually attach to. I observed crabs also, smaller ones, but I still observe crabs in the 3 beds with the highest densities. So I'm thinking maybe the thicker beds are better suited for more mobile intertidal species rather than gastropods. Some sources of error or limitations, I could have accidentally missed out on a holdfast or a gastropod. Some of the beds were pretty thick. I did my best to try not to miss them, but there's always the possibility of human error there. I also only did 6 replicates, and the quadrat was pretty small, and I did halves, and they were only 50 centimeters by 50 centimeters, so maybe having more replicates would give us a more accurate result. Another source of error was initially when I misidentified Fucus spiralis as Ascophyllum nodosum, but I ensured it was Fucus spiralis in all my quadrats, but initially I did misidentify it. I only performed the study in one region, which was the urban rocky shore area, which was Point Pleasant Park. And so maybe for future recommendations, I would ask maybe more research needs to be done regarding the relationship between gastropods and Fucus spiralis, specifically, as well as expanding the research beyond tropical and mangrove areas, which is where I found most of the articles focused on. I think it might also be a good idea to conduct more in-depth studies on the distribution of gastropods within the tidal zone with the abiotic factors I mentioned previously, perhaps to determine if these factors are more influential than the density of vegetation. I think incorporating the idea of two-eyed seeing is important as well when conducting fieldwork, because it enhances our relationship with the organisms and the environment we're studying. I read about an example incorporating two-eyed seeing with science, and it actually happened in Nova Scotia. It is a collaborative environmental planning initiative to protect watersheds in a specific area, and it incorporates traditional Mi'kmaq and Western perspectives, and one of their goals is to balance environmental, social, and cultural objectives to ensure the health and sustainability of the watershed system. I don't often see science-based initiatives prioritizing social and cultural objectives equally with environmental objectives, because from what I've experienced as a marine biology and sustainability major, unfortunately, Indigenous teachings and values are often an afterthought or not incorporated at all. So I thought that was pretty interesting, and I included that in my references section if anyone's interested in it. I think by approaching research or science from a two-eyed seeing perspective, we can obtain and develop more accurate goals and recommendations based on our results in a more holistic way and in a manner that is more directly connected to the environments we are studying. And so for the final conclusion, the gastropod densities were found to be negatively correlated with Fucus spiralis, and actually predicted they'd be positively correlated, so I was surprised. Here are all of my references, and if anyone has any questions or wants to talk further about it or wants more details on the references, just ask and I'd be happy to answer. Thank you.